اوكي شباب صبايا اليوم راح نكمل ان شاء الله محاضرتنا الثالثة. The title of this lecture is Secretory Functions of the Gastrointestinal Tract. We will talk mainly about the salivary and gastric secretions. Now, the secretory glands within gastrointestinal system serve several functions, most importantly the secretion of the digestive enzymes to digest the food particles, and these glands present from the mouth to the distal end of the ileum. On the other hand, there is another kind of glands that secrete mucus for the lubrication and protection of the gastrointestinal mucosa, and these glands spread from the mouth toward the anus. A few words about the types of the secretory glands within gastrointestinal system. The first kind of gland is single cell mucus glands, or simply called mucus cells or goblet cells. They're called goblets for the appearance of the cell, and these cells secrete mucus to the epithelial surface, and the mucus is important for the purposes of lubrication and protection of the gastric mucosa. Mucus is really important for the function of the gastrointestinal tract. It is a kind of thick secretions, and it's composed of water, electrolytes, and some kinds of glycoproteins, and it gives lubrication and protection for the gut wall. And the mucus has several characteristics. One of them, it has adherent qualities. It coats or cover the gut wall to prevent contact of food with the mucosa, so it gives a protection function. It is also help in sliding of food. Also, it helps in adhering the fecal particles together to form the fecal matter. It provides resistance for the digestion by the gastrointestinal enzymes, and the glycoprotein components of the mucus are important buffer for acids and alkali. The other type of secretory glands within the gastrointestinal system is the pits, and they represent invagination of the epithelium into the submucosa. Within the small intestine, they are called crypts of Lubberquin. This is an example of the crypts of the small intestine. The third type of secretory glands within the gut are tubular glands, and these kind of cells secrete acid and pepsinogen. An example of these are the oxantic glands located within the stomach and the duodenum. And this is the structural histology of these glands. The fourth type of the secretory glands within the gut are the complex glands. Examples on these are the salivary glands, the pancreas, and the liver. These glands are compound, or mainly the salivary and the pancreatic ones, are compound acinous glands, which means that they are compound of the acinous part plus the ductules. Now, the gland function within the gastrointestinal is controlled by several mechanisms. The first one is local, where there is a tactile stimulation of the mucosa by either distension or irritation, resulting in controlling either increase or decrease the secretion of these glands. Also, nervous stimulation would control their function. Also, hormonal mechanisms are very important in controlling the gastric secretions. Or not only the gastric, any glands within the gastrointestinal tract actually is regulated by gastrointestinal hormones, mainly by the stomach and intestine. The mechanisms are mainly regulated within the stomach and the intestine to control the gastric and pancreatic juice secretions. Talking about the role of the autonomic nervous system in controlling the glandular function, the parasympathetic nervous system increases the rate of the glandular secretions, and this is conveyed through the glossopharyngeal and the vagus nerves to the salivary, esophageal, gastric, pancreatic, and duodenal glands. The pelvic nerve also carry parasympathetic nerve fibers to the glands of the large intestine. The sympathetic has dual effect, either increase or decrease. The decrease of salivation mainly induced by the vasoconstriction of the blood circulation. Now the mechanisms how is the organic substances, water and electrolytes are secreted by the gastric glands. Firstly, the supporting or the nutrient substances coming from the circulation at the base of the cell, from the capillary, and there at the base of the cell there is a lot of mitochondria, which is important for the production of the ATP required for the synthesis of the protein at the endoplasmic reticulum and the ribosomes. So these proteins produced by the ribosomes fuses with the Golgi complex apparatus here to form secretory granules or vesicles. These vesicles will stay in the cell until there is a hormonal or neuronal stimulation for the release, and the release is mainly excited by calcium entry to the cells by the process of exocytosis. It's important that the neuronal or hormonal stimulation will result in passage of water and salt through the glandular cells to wash any organic substances through the secretory border to the lumen at the same time of secretion of the organic substances. Now we will start talking about the salivary glands in the gastrointestinal system as the first part. And there is 3 major glands called the parotid gland, the submaxillary or the submandibular gland, and the sublingual one. Also there is other small glands including the buccal glands, which are distributed within the oral cavity. Now the secretion of the saliva has 2 types. The first type is serous secretion, which is watery in nature, and this part of the saliva contains the alpha amylase enzyme. So it's important to know there is enzyme called alpha amylase or ptyalin within the serous secretion of the saliva, and this enzyme is critically important for the starch digestion. As mentioned in the last lecture, starch is not digested in the human cellulose is not digested in the human body, and the process of starch digestion is started within the mouth through the salivary secretion. The other type of secretion is mucus, and this contains mucin, and it's important for the process of lubrication and protection of the gastrointestinal wall. So the parotid gland secretions are mainly serous, while the buccal glands are mainly mucus. The submandibular and sublingual glands produce mixed, both serous and mucus secretions. The maximum rate of secretion of the saliva is 4 ml per minute, and the secretion of the saliva is decreased during sleep, between the meals, and the average daily secretion of the saliva is about 1 liter of the saliva. However, this comes up to 1.5 liter per day depending on the requirements, and the pH for the saliva is 6 to 7, and this pH is really important for the work of this enzyme, the alpha amylase. So any pH below or above this level would result in improper work of this enzyme, so no starch digestion will take place properly. Now the functions of the saliva, several functions. The most important one is lubrication, solubilization of the dry food, and oral hygiene, because the saliva has many contents that have bacteriostatic, that kills the bacteria, such as thiocyanate ions. Also it has some lysozymes that damage the bacteria, and also it has antibodies that has antibacterial effect. As I said, that there's digestive function for the saliva through the salivary enzymes, and this amylase has been mentioned many times before, and this enzyme is actually stimulated by acid medium that digests starch into maltose. There is other enzymes within the salivary secretion such as maltase, and this enzyme is secreted by the major salivary glands that digest maltose into glucose, and the lingual lipase, which is involved in the digestion of triglycerides, especially in the milk, into fatty acids and diglycerols. Now, the saliva is secreted at two stages. The first one as the acini, at the acini part of the salivary glands, and this primary secretion is the component of these secretions are similar to that what's present in the plasma, and it contains the enzyme taline or alpha amylase, mucus with extracellular fluids. Once these secretions move to the ducts of the salivary glands, there will be modifications. There will be absorption of some ions, secretion of other ions to modify the component of the saliva. So the first ion to be reabsorbed actively is sodium, and this would result in minus 70 millivolts osmolarity of the saliva. This would result in passive absorption of chloride ions from the ducts. Also, potassium is actively secreted to the ducts, and bicarbonate either passive or active secretion of these ions to the ductules. Now, the ionic compositions of the saliva depends on the rate of secretion. And at the resting conditions, the sodium and the chloride concentrations equals to 1/7 to 1/10 of the plasma concentration. The potassium ions concentration is 7 times as much as the plasma concentration, and the bicarbonate ions concentration is 2 to 3 times as the plasma. Saliva is always hypotonic, and during the maximal salivation, when there is increase in the salivation excretion, this rate could be increased by 20 times, and this will result in changes in the ionic component of the saliva, increasing the osmolarity. So the sodium and the chloride ion concentrations will be about 1/2 to 3/3 of the plasma. The potassium concentration will be reduced, so this will come from 7 to 4 times as the plasma. And it's important to know that loss of saliva from the body can lead to hyponatremia or potassium depletion. This picture shows summary of the components of the saliva. As you can notice that most of the saliva is water. 99% of the saliva is water. These kinds of enzymes present within the saliva within organic substances. Also, there is other proteins, immunoglobulins, non-protein compounds, all these present in the saliva. The ionic components, and there are some gases within the saliva. Normally, the glucose is not present in the saliva. However, patients with diabetes mellitus, مرضى السكري بيكون في عندهم glucose is detected within the saliva. However, normally it should not be present within the saliva. Now, this diagram shows how the rate of excretion would affect the components. If you look at the sodium, more increase in the flow would result in more concentration of the sodium. So the sodium would not be absorbed effectively as in the resting conditions. The chloride is also increased concentration. Like the potassium, usually not that much difference. However, it's reduced, and the bicarbonate concentration is slightly reduced as well. Now, the nervous regulation of the saliva is controlled by both sympathetic and parasympathetic nervous system. Talking about the parasympathetic nervous system, coming from the superior and inferior salivary nuclei in the brainstem, that is excited by any stimuli from the tongue, from the mouth, from the pharynx, and even any appetite area in the central nervous system will control the salivary process. So it's important to know that the superior and inferior salivary nuclei are the controlling centers for the salivation, in addition to appetite center within the central nervous system. Now, the kind of taste will increase or decrease the salivation process. For example, any sour taste will increase the salivation, and this could be increased up to 20 times fold. Smooth objects within the mouth will increase salivation. On the other hand, rough objects will decrease the salivation. Also, reflexes in the stomach and upper small intestine, for example, irritation or even nausea, will result in increased salivation rate. Now, the sympathetic nervous system also increases the salivation. However, its effect is weaker than the parasympathetic nervous system. And the sympathetic nervous system originates from the superior cervical ganglia, and it travels through the surfaces of the blood vessels into the salivary glands. Also, the parasympathetic nervous system will increase the blood supply to the glands, resulting in vasodilatation, and this is a direct effect will result in increased salivation. So the parasympathetic works directly on the salivary glands, increases its secretion. It also works on the blood vessels of the salivary glands, resulting in vasodilatation, and this vasodilatation within the salivary glands will increase the process of salivation. On the opposite way بتصير عملية عكسية. كل ما زاد الsalivation بصير في عنا process of vasodilatation by a process of generation of bradykinin, which is a vasodilator catalyzed by or produced from the alpha-2 globulin within the circulation, adding more effect to increase salivary secretion. Now, a few words about the esophageal secretions. The esophageal secretions are minimal. هي مهمة only for the lubrication and passages of food from the upper gastrointestinal system to the stomach, and it's mainly mucus. Different kinds of glands in the main body of the esophagus. We have simple mucus glands, while in the gastric and initial portion of the esophagus is compound mucus or are compound mucus glands. As I said, it's for application and protection of the mucosa from food and acid reflux. Now, talking about more important secretions within the stomach, the gastric secretions. The stomach has three types of glands. The first one is the mucus glands, and they secrete mucus. The other type of the glands are the oxyntic or gastric or parietal glands. These kind of glands, the parietal cells, which secrete the intrinsic factor and the hydrochloric acid or the gastric acid, and the chief cells component of these glands secrete pepsinogen. These are located inside the surface of the body and the fundus, and they represent the proximal 80% of the stomach structure. The pyloric glands, which are located at the distal 20% of the stomach, they secrete the mucus from the mucus cells and gastrin from the G cells, and gastrin, it is secreted to the circulation. This is a hormone, will go to the circulation and cause a distal effect. Now, the gastric acid is important in three major functions. The first one is bacteriostatic, so it will kill the bacteria. It's important for the activation of pepsinogen into pepsin. We will talk about that later on. And it is important to begin the process of protein digestion, mainly using the enzyme pepsin. More words about the parietal cells of the gastric mucosa. So the gastric juice contains about 160 millimole per liter of hydrochloric acid, and this is isotonic with the body fluid. Now the pH of the gastric secretions is very acidic, is about 8.8, and this is very, very acidic. And the ion concentration, the hydrogen ion concentration with the gastric secretion is 3 million times as the arterial blood, so it's very acidic. Now the acid, the HCl of the gastric secretions is formed at the villus-like structures of the canaliculi within the oxyntic or the parietal cells, and it is secreted to the mucus. Just bear in your mind عشان كمان شوي راح أذكركم فيها إنه ال HCl is secreted to the lumen and the bicarbonate is absorbed at the basolateral membrane into the circulation. HCl is secreted and bicarbonate is absorbed. Now, the mechanism of HCl secretion فقط هي شوي تركيز. المعلومات جدا بسيطة في هاي المرحلة. بدنا نعرف كيف الأسيد is secreted into the lumen of the stomach. First of all, if you look here, water inside the parietal cells. So just to be oriented, this is the parietal cells, and this is the lumen side, and this is the circulation or interstitial fluids. The blood is here. So water inside the parietal cells will dissociate into hydrogen ions or proton and OH. Okay? Now, these hydrogen ions will actively secreted into the lumen in exchange for potassium ion. So hydrogen out, potassium in, using this hydrogen potassium ATPase. This is the second step of HCl secretion. The third one is at the basolateral membrane, potassium is transported into the cell in exchange for sodium, which is transported outside the cells using sodium potassium ATPase. Okay, after this step, the potassium which is transported inside the cell will leak into the lumen. But this leak will be, or this potassium leaked out from the cell into the lumen will be actively transported back to the parietal cells in exchange with proton using the proton potassium ATPase. Now, at the basolateral membrane here, because potassium is transmitted inside and sodium is exported outside, this will result in low intracellular sodium. So sodium will come from the lumen of the canaliculi into the cell. And at this stage, because water dissociates into proton and proton is excreted outside the cells, we have the OH here, which combine with carbon dioxide to form bicarbonate using an enzyme called carbonic anhydrase. So OH combined with carbon dioxide forming bicarbonate, and bicarbonate is excreted outside in exchange with chloride. So bicarbonate is out, chloride in. Now this chloride actually will leak into the lumen. Chloride will leak into the lumen by diffusion. So we have chloride here, we have hydrogen ion here that interact with each other forming HCl, hydrochloric acid, And this is how is the HCl is formed inside the lumen of the. Now the last thing to move is water, and this will move by osmosis. Water will always move by osmosis. This is as simple as that. Proton is excreted to the lumen, chloride is also excreted to the lumen, and bicarbonate is absorbed to the circulation. And these are the osmolarity of the hydrogen concentration. The potassium concentration is about 15 milliequivalent per liter. The proton is 155 milliequivalent per liter, and the chloride is 173 milliequivalent per liter. Now, this is just a summary of the gastric juice. As we said, there is, as in saliva, water is the major component, and there is some enzymes. Actually, you have to notice that this is renin is not present in the human secretions. We have other enzymes, gelatinase, urease, mucus, and intrinsic factor. We'll talk about them in more detail, and these the ionic components. Now, the secretion rate will also affect the ionic combustion. So, at low secretion rates, which always present between the meals, there is high sodium chloride. However, after meal, high HCl. And always the gastric juice is isotonic. And this is the graph you see. As much the rate of increasing the rate of excretion would result in increase the HCl, while the sodium chloride will reduced. Now, the gastric secretion is regulated by nervous or hormonal mechanisms. The acetylcholine will stimulate the HCl secretion, hydrochloric acid secretion. It will also stimulate mucus, pepsinogen, and gastrin secretion. Histamine mainly work on HCl secretion. Gastrin as well increase the HCl secretion, and it's more potent than histamine inducing HCl secretion. Now the other enzyme is pepsinogen, secreted by the gastric glands, and as its name indicates, anagen by the end of a protein or precursor mean is inactive and needs to be activated. This pepsinogen is activated by HCl within the stomach that converts pepsinogen into pepsin, and the pepsin will work thoroughly to convert more pepsinogen or to activate pepsinogen into pepsin. And it's important proteolytic enzyme that working at 1.8 to 3.5 pH. Every enzyme has its optimal pH, so any pH above or below this level will result in improper action of this enzyme. This enzyme is reversibly inactivated at pH higher than 5 and irreversibly inactivated at pHs higher than 7 to 8 values. Now the intrinsic factor, which is secreted by parietal cells, is important for the absorption of the vitamin B12 in the ileum. And when there is destruction of these parietal cells within the stomach, for example, there is ileal disease, there is a surgery, there is atrophic gastritis, chronic inflammation with the gastric mucosa, this would result in achlorhydria, means that there is no HCl secretion, and pernicious anemia. Pernicious anemia results from the failure of maturation of the red blood cells because there is no vitamin B12. Now, in the stomach, the dietary vitamin B12 interact with a binding protein present within the gastric juice, and at the duodenum level, the pancreatic proteases digest that binding protein, so the B12 will be released and interact with the intrinsic factor at the ileum. This vitamin B12 intrinsic factor complex will be absorbed. So intrinsic factor is important for the absorption of B12, and defects in this process will result in pernicious anemia. Now, the pyloric glands, they are structurally similar to the exanthic glands. However, they have fewer peptic cells with no parietal cells, and they contain mucus cells mostly. These cells also secrete small amounts of pepsinogen, however, large amounts of thin mucus, and this mucus is important for the process of lubrication and protection of the stomach wall from digestion by gastric enzymes. Also, the pyloric glands secrete gastrin, and this is an important hormone for controlling the gastric secretion. Now, a few words about the gastric barrier. The surface epithelium of the gut secretes mucus, and this mucus thick is about 1 ml thickness. It is alkaline viscid mucus. This mucus, in addition to the tight junctions present between the epithelial cells, protect the stomach mucosa from digestion by gastric juice because it's very highly acidic secretion. So the mucosa is protected by this mucus barrier. In certain occasions, like ingestion of aspirin or alcohol, this barrier is damaged, resulting in digestion of the gastric mucosa by the gastric juice, and peptic ulceration takes place. Now, the regulation of the gastric acid secretion is taking place by two different mechanisms, either by nervous control or hormonal mechanisms. So if you look at this picture, at the vagus nerve, secretion of the acetylcholine will stimulate the parietal cells to secrete hydrochloric acid. Also, the vagus nerve stimuli for the gastric cells will result in release of the gastrin into the circulation. This gastrin go through the circulation to the parietal cell, secreting more or stimulating the cells to secrete more HCl. Also, local reflexes within the mesenteric nervous system results in release of acetylcholine that's stimulating the G cells to produce gastrin, and also amino acid components of the gastric chyme will result in the secretion of gastrin. في عنا structure مهم بالأوكزنتيك glands called enterochromaffin-like cells or ECL cells. These important cells are stimulated by gastrin, and the secretory product is histamine. Histamine works on the parietal cells, resulting in stimulation of these cells to produce more hydrochloric acid. So ACL cells produce histamine and it's stimulated by gastrin. Now, pepsinogen secretion is stimulated by acetylcholine, which is released either from the vagus nerve or enteric nervous system, or the presence of acid in the stomach. And this is indirect effect mediated by enteric nervous system reflexes. Now, there is other enzymes within the gastric secretions which were not mentioned, and there is a kind of enzyme called gastric lipase and gastric that is involved in the digestion of triglycerides to form fatty acids and glycerol. There is another enzyme called gastric amylase. Please pay attention, this one is different from the salivary amylase. The salivary amylase is more potent, stronger in the digestion of the starch. This enzyme has a very minimal effect, and it digests starch into dextrin and maltose. The gelatinase is an enzyme involved in the digestion of gelatin and collagen of meal to produce peptides, and urease is important in the digestion of urea to produce ammonia. Now, the gastric secretions are regulated by three different phases depending on the presence of the food. The first one is the Cephalic phase, where there is no food inside the mouth actually. You're just seeing, smelling, thinking about food. There will be a stimuli within the cerebral cortex and the appetite centers going through the vagus nervous system to the stomach, asking the stomach, please start secreting your acidic juice. And this phase is important in producing about 30% of HCl secretion in response to the meal. The stimulations act through either mechanoreceptors or chemoreceptors, the smell and taste receptors, either central pathways like thinking about the food, and hypoglycemia. So hypoglycemia would result in increasing the gastric acid secretions. Since the vagus nerve is the carrying nerve for the stomach or the nerve that tells the stomach to secrete the acid juice, no vagus nerve, so there is no cephalic phase of gastric secretion. So vagus nerve is important, and this effect is diminished by vagotomy. The gastric phase happens when the meal is inside the stomach, and this is responsible for the bulk secretion of the gastric acid. About 60% of acidic secretion induced by this phase, and about 1.5 ml 1.5 liters of gastric juice secreted by this phase. And the stimuli is by vagovagal reflexes from the stomach to the brain and then back to the stomach, or either local enteric nervous system reflexes, and finally through the hormone gastrin. The third phase, or the last phase of gastric acid secretion, is the intestinal phase, and this is responsible for about 10% of acid secretion in response to the meal. This phase is stimulated by the presence of food in the duodenum, and there will be small amounts of gastric juice secreted, and it is mainly stimulated by the hormone gastrin, which is secreted by the duodenal mucosa. This picture summary actually the phases. It's the same thing I've said, that there is conditioned and unconditioned reflexes like the sight, the smell, hearing about food, or even the presence of food in the mouth will stimulate the vagus nerve, resulting in stimulation of the gastric juice. This is the cephalic phase, only nervous. The gastric phase is both nervous and hormonal. Bolus, or the presence of food in the stomach, will stimulate either the vagus nerve, the local nerves, and gastrin will result in stimulation of the gastric juice, and this represents the major or the bulk of the gastric secretions. The intestinal phase is mostly hormonal, stimulated by gastrin and inhibited by secretin, cholecystokinin, somatostatin, gastrointestinal peptide, and VIP. These inhibits the secretion of gastric juice. There is other intestinal factors that inhibit the gastric secretions or slow the secretion rate. The enterogastric reflex, which is transmitted through the myenteric nervous system of the gut, the sympathetic and the vagus nerves, and this will result in or lead to decreased stomach secretion. This reflex is stimulated by distension of the small intestine, acidic material in upper intestine, protein breakdown of products or irritation of the mucosa. Also, any dietary breakdown products such as acids, fat, protein, hyperosmotic or hypoosmotic fluids in the upper intestine, or any irritation in the upper intestine will stimulate secretin, and this enzyme will result in controlling the pancreatic secretions. We'll talk about the pancreatic secretion next lecture, inshallah. And this will result, secretin will result in decreased stomach secretion. Glucose-dependent insulinotropic peptide or gastric inhibitory peptide, GIP, vasoactive intestinal peptide, and somatostatin reduce also the stomach secretions.